Thank you so much for listening to American Medicine Today. I'm with my great group of friends, Ethan Euchre.
Pleasure to be here.
Jeff Wagstaff.
Hello, everyone.
Hello. And world-renowned orthopedic surgeon, Dr. Alfred Bonatti.
Hello.
Now, nearly a year after the media flurry surrounding its controversial launch, a company called Nectome is revealing two pieces of news. And joining us to discuss is Robert McIntyre, founder and CEO of Nectome, a neuroscience research company dedicated to advancing the science of memory preservation. Thank you for being here, Robert.
Thank you, Kimberly. It's a pleasure to be here with you.
Yes.
Now, I was mentioning this to the group off air before we went on. I actually reached out to you, Robert, over a year ago. Well, not to you personally, but to your people. And it's because I saw, and we want to get this out of the way at the top of the interview and let you clear things up. The article I saw was regarding a preservation of the brain that was essentially 100% fatal. And it was, I guess, the way the article was written, you were injecting chemicals or a fluid into the brain that was supposed to preserve the brain for a later date, similar, I guess, to sort of cryogenics, but it was going to kill people. And the media pounded you over it. Just talk about that a little bit and how you kind of switched gears and what you're doing now.
What I really regret is kind of derailing the conversation on speculation about what this technology looks like in the future, because what we're doing right now is we're trying to build technology that can preserve long-term memories that are stored in the brain. What we're actually doing is basic research to create this technology. So I think it's very important. I think that being able to preserve memories is a profoundly important technology, and the media sort of got ahead of itself and focused a lot on the speculation and less on what we're actually doing right now.
So what is the primary goal of Nectome's research then? You're trying to preserve it, but how are you doing so?
The goal is to be able to archive long-term memories that are stored in neural tissue. And so I think the key point to think about here is that long-term memories literally physically exist in your brain as anatomical changes. The brain can't create a long-term memory other than by actually changing its nanoscale anatomy. And one of the really important consequences of that is that it may very well be possible to preserve your memories, to preserve information that's stored in the brain.
How can you measure that? Because you don't have any background on. I mean, at least I don't know. That's what I'm asking the question. Is if you are going to preserve a memory, you need to have a background of that animal or that individual that has something that you can bring it back and evaluate. How you do that?
There's a few things I think about with that. First of all, we've known for quite a long time that long-term memory survives electrical blackouts. So even in the absence of electroactivity, long-term memory persists. For example, if your heart stops beating within 60 seconds, there is no electroactivity in the brain, and yet people can survive several minutes of ischemia, no blood flow, and it does not erase long-term memories. They've got to be built out of something fairly robust, or they wouldn't survive these types of things. And what we've seen in neuroscience is that you generally have changes in synapses to create a long-term memory. So, I mean, literally these tiny little connections between neurons will actually change size when memories are formed. And that's how these memories are able to be so robust, because it's an actual nanoscale anatomical change in the brain. And one of the interesting consequences of that is that if you can preserve those, that is at least the first step that you need to do for preserving memory. This is why several years ago, I think 7 years ago at this point, Ken Hayworth, who's a neuroscientist, created this prize called the Brain Preservation Prize for the first team that could preserve all of the synaptic connections in a brain in exquisite detail. And that's what really got me into this whole field, is this prize. I was very excited about it, and I thought I could actually pull it off.
If you have the synapses and all these different things that were, I mean, does it really preserve, though, the memory or
just the tissue or the neurons?
First, there's an analogy, and there's also the work that we're currently doing with extracting long-term memories from preserved organisms. The analogy here is, I think, with DNA. In the 60s, if it was 1960, we very much understood what DNA was. We understood that it is the molecule of heredity, and we knew chemically how it was held together. And you could make an extremely compelling argument that you can preserve DNA if you use liquid nitrogen. And so what can you do with the DNA in the 60s? Very little. You could maybe scan a little bit of it, but the idea of scanning an entire gigabyte worth of data, a whole genome, was incomprehensible in the 60s. And yet, let's say you have a species in the 60s that's about to go extinct. You have the last instance of the species right here. Do you throw its DNA away, or do you preserve it with a technique that you're very confident does work to preserve that information, even though you can't access it right then and there? And I think there's a very good argument to be made for preserving the information. If you had done that in the 60s, yeah, you're not going to get it back in the 70s or the 80s or the 90s, but in the early 2000s, we had the Human Genome Project. And now today, what would have cost $1 trillion in the 60s to scan a whole genome now only costs $1,000. It only takes a day to do it.
Wow.
So if you had these preserved genomes from the 60s, you could actually scan them today. But to address this question of long-term memory, the research that we're currently doing now involves extracting a single bit of information about long-term memory from preserved worms. And so that's the current work that we're doing right now, is preserving these tiny little animals and then extracting information about their long-term memories afterwards.
Robert, how would we know if a worm even has memory, let alone preserve said memory?
What we focus on right now is tap habituation. These worms live in these little petri dishes, and they crawl around on the surface of the dish, and they eat bacteria, and they have a good time. But if you were to take your finger and flick the petri dish, give it a little mechanical tap, it makes a little earthquake, and the worms freak out. Some of them dart forward, some of them turn around back on themselves and then kind of go away. They respond to that tap, similarly to if they'd run into something while they were exploring. That's an aversive response to that tap. But if you tap the plate over and over again at repeated intervals, they eventually get used to it, and they don't respond to it hardly at all. It's kind of like if you live next to a train station. You just moved into your house, and you may get surprised when the train goes by. But after you live there for a month or two, you sort of don't even hear the train anymore.
It's like conditioning, basically.
It's not important, exactly.
Yeah.
And so that's habituation. And this lasts. Even if you tap this dish and you wait 24 hours and you haven't done anything, the worms that were habituated still don't really care if you tap that dish. And so the question is, if I had 100 worms, right, and 50 of them were habituated and 50 of them were naive and they were alive, you could just tap them and you could see the ones that react a lot are naive and the ones that don't are habituated. But let's say we preserve them with this fixative. Then the question is, can you look at their nervous system and can you still say, anyway, these were the ones that were naive and these were the ones that were habituated? If you can get that right, then that's one bit of long-term memory that you've extracted.
When you look at the overall project of memory preservation, are you doing it internally? For example, if you were to do it with human subjects, would you be helping them retain their memory? Or are you trying to almost like download their memory externally?
We're just focused on preserving memory and validating that the preservation works. That's where we're at with long-term memory right now.
You just said before, and I know that one because that's exactly what happened with the people who die in ICU, and then later on they come back and say, "Oh, I saw a light," and that's what, and it's being religious in one way, but I really think it's electrical. Now, if you do something like that in the brain of somebody who just died, and then you have the opportunity to see that probably is going to be active electrically for a few minutes, can you do that instead to be doing in worms? Can you do that in that human brain?
Where we're currently at is we've been able to preserve rabbit brains in very great detail, and we won a prize for that. We can preserve all the synaptic connections. And we scaled it up, and we're also able to do an entire pig brain as well, which is about 10 times as large. We won that prize last year, and now we're focused on extracting memories from worms, because their nervous system is so simple you can actually analyze it and sort of interpret what it means. And so that's where we're focused right now, and I think there is a clear progression for extracting more bits. Right now we're doing one, but you could certainly imagine a paradigm where you get two out of a worm, and you could get 4. And where that's headed is a more comprehensive ability to access information that's stored in brains. I don't know if that answers your question.
Not really. That's fine. That's fine.
But this is all sci-fi-esque.
Yeah.
So it's so interesting to see, where science is going.
Well, and I like the way what you're saying, Robert, is basically, especially when you keep referencing what they did with the human genome in the 60s, is you're saying what you're working on now, and you don't want to speculate about the future, I know that, but what you're doing now will be useful in the future.
It's really important to think about the ethics right now, and that needs to be done before this technology is ready. So what I'm really excited about is this new ethics organization that's being put together by Anders Sandberg at the Future of Humanity Institute, and they're thinking about exactly these questions. What does it mean to preserve memory? You know, what is the relationship between one generation to another if you're able to preserve long-term memories and have a sort of living history instead of just a written history? And those are really important questions to be thinking about. And we should be thinking about them now and not once the technology is actually ready to go.
Good point.
I agree. Thank you so much, Robert McIntyre, founder and CEO of Nectome, a neuroscience research company dedicated to advancing the science of memory preservation. Thank you so much for being on the program.
It's a pleasure to be here. Thank you.
Really hard to wrap your head around some of
I
that
know.
stuff, at least for me.
You know, we have computer-brain interfaces and all these things that maybe you read about in sci-fi novels, and then it's coming to fruition. So it's just an interesting technological advancement. Make sure you stay tuned. You're listening and watching American Medicine Today.